I'd now like to introduce Professor Ben Hayes from QAAFI and the University of Queensland, just to give us a bit of genomics lesson. So thanks, Ben. Cheers.
Okay, good afternoon, everybody. The theme of my talk, I think, is if you're going to multiply or scale up embryos, you better pick the right ones to scale up and multiply. So just to lead into that, another little genetics lesson. It's a little appreciated fact that there's as much genetic variation within families as there is across families. Another way of saying that is if you mate a sire and dam, you get lots of progeny from that sire and dam. There's as much variation in that cohort as there is across the different families. And if you're in the AI business, you probably sort of live this pain quite often because you can have an outstanding sire, an outstanding dam, you put them together and you get a crappy bull that never makes the cut. The flip side of that is you can get an outstanding bull. Why is that? It's because those sire and dam, although they're outstanding, they can actually carry really good chromosomes that improve traits like beef production, marbling, fertility and so on. But even the very best sires and dams also carry some poor chromosomes. And when you get that crappy animal from that good sire and dam, that's what's happening. Just by chance, they've got that random sample. So if we shift that to the embryo side of things, when we put our really elite sire and dam together to start producing our embryos, what we want to see is which of the embryos has got the good stack of chromosomes from the sire and dam. And we want to discard the ones that have got the poor stack. So how do we do that? Well, that's the technology we've been working on. So it turns out from an embryo, just like an animal, you take a tail hair, an ear punch, extract the DNA. From an embryo, it's a little bit more tricky. You've got to do a biopsy. You're dealing with much smaller amounts of DNA, but the idea is the same. And you can run that through a sequencer or a SNP chip, and you can produce genomic breeding values for that embryo. And that is a very good way of working out which embryos to focus on. So you can think about the range of traits you want to do that for, and that comes back to the breeding objective or the product that you want to breed for. That can include things like meat quality, fertility, growth, and so on. Just to give you a concrete example of this, we worked with Nbryo over the past couple of years to take some of the embryos that were produced. We extracted DNA from biopsies for those embryos, and then we ran that DNA through a sequencer that I'll explain in a moment. We pulled out of that the DNA that was relevant to our genomic predictions, and these were the Northern Genomics Project GBVs that you might have heard about. So heifer puberty, tick resistance, buffalo fly lesion resistance, and so on. So we were able to get GBVs on those embryos. In the future, we're going to be able to do the same thing for things like eating quality, consumer eating quality. Nice picture back here. We're also going to be able to do it for methane. So right at the moment, we have a large MLA project where we're running animals through a device that assesses how much methane they're emitting. We're up to about, I think, nearly 800 animals that have run through that. And that is our basis for starting to calculate these genomic breeding values for methane. So that's coming down the pipe. And all this can be done on calves from a tail hair or TSU. It can also be done in this embryo cycle for scale up and multiplication. So you really do have the opportunity to choose pretty precisely which genetics you want to amplify up to become the future herd. Okay, the other part of this is what this picture's about. When you do this, you really want to do it as quickly as possible. If you think about, you've got your embryos sitting there, you want to make decisions about which ones you're going to keep or discard, which ones you're going to focus on really quickly, because you've got to implant them and so on. So you've got to have a quick turnaround time. And the next sentence sounds a bit strange, but this is where we were inspired by COVID. So during the COVID epidemic, many of you may have noticed the first time you got a COVID test, stuck the thing up your nose, it got sent away, and was it 48 hours or something before you got your result back? So you had to stay at home and not go anywhere. By the end of the COVID epidemic, that was all happening in about half an hour that you got your result back. And it's because of the little machine you can see in the picture there. That thing that guy's holding, that's actually a genome sequencer, a mini iron. And that's exactly what we use as well. So you can put your DNA in there, and within minutes, you get the DNA of the animal or the embryo or whatever you've got in there starts coming out onto your iPhone or onto a computer. And then you can turn that into genomic breeding values and start to make decisions about what you're going to do with that embryo. So this is what we're working on. The stage we're at, I told you the example of calculating genomic breeding values on these embryos. We actually did it with this machine, the handheld machine, but it took us about 6 hours or so to turn it around and to do the data analysis to crunch into genomic breeding values. So we want it a bit quicker than that. It's all about getting that DNA extraction right and so on. So that's where we're headed. The other thing I will say is embryo quality, of course, is really important. The embryo is constantly putting out signals about how healthy it is. It's switching genes on and off, and if you know how to read that off, you get a profile of how healthy that embryo is, and perhaps how likely that embryo is to turn into a calf on the ground and so on. So together with Nbryo, we're actively exploring those signals. That's been used in human genetics quite a lot. We're translating that technology into cattle. So that's just a snapshot of where we're heading.